Welcome to this explainer. Go ahead and look down at your hands for a second. Take a deep breath. You are currently piloting the ultimate architectural marvel, the human body. But to truly understand the big picture of how we're built, we really have to look through the lenses of some highly specialized branches of science. So today, we're taking an incredible journey into the human blueprint. We're going to start by looking at the broad, massive, macroscopic structures that make up our bodies, and then we're traveling all the way down into the microscopic origin story of human life itself. We're talking about how embryology fits into this larger context of specialized branches. It's a fascinating progression, so let's just get right into it.
All right, let's start with the foundational science that makes sense of our physical form, anatomy. The word itself comes from the Greek roots ana, meaning up, and temnein, meaning to cut. Historically, anatomists literally had to cut structures apart just to figure out how we were built. And honestly, this structural framework is absolutely vital. Think of it kind of like a car engine. You simply cannot understand how the engine works, the physiology, without first understanding what the parts are and where they go. That's the anatomy. In biology, form always dictates function. Now, to decode the human body, scientists generally split anatomy into two major perspectives. It's almost like putting on two completely different magnifying lenses used by specialized branches to understand our design. On one side, we have gross anatomy, or macroscopic anatomy. This is the study of large structures you can easily see, like, holding a heart in a dissection lab, or looking at a whole brain. On the flip side, we have microscopic anatomy. This is an incredible shift in scale. It requires actual magnification to see what's going on at the cellular level, which is cytology, or at the tissue level, which is histology. These tiny microscopic elements are the foundational building blocks that actually assemble those larger gross anatomical structures. So here's our roadmap for this explainer. We'll start with the branches of human anatomy, then dive into embryology, the origin story. Next up is the third week of gestation, followed by forming the body cavities, and we'll wrap up with when development goes wrong.
Let's kick things off with Section 1, Branches of Human Anatomy, Mapping the Body. When scientists are tackling the sheer vast amount of structural data in gross anatomy, they usually take one of two main approaches. The first is regional anatomy. This is where you look at absolutely everything packed into one specific zone. Imagine a trauma surgeon operating on the abdomen. They need to know exactly how the muscles, nerves, blood vessels, and organs all interrelate in that exact physical space to do their job safely. The second approach is systemic anatomy. Here, instead of focusing on a region, you trace a single functional system throughout the entire body. A cardiologist, for instance, uses systemic anatomy to follow the cardiovascular system, tracing blood flow from the heart literally all the way down to the tips of your toes.
Moving to Section 2, Embryology, the origin story, bridging the gap.
We've just mapped out the specialized branches that deal with the finished human body. But how do we actually get there? How does microscopic anatomy become macroscopic gross anatomy? Well, embryology is, quote, the science of the development of an embryo from the fertilization of the ovum to the fetal stage. It is the ultimate biological prequel. It's the dynamic, step-by-step process that explains how we go from just a couple of microscopic cells into a fully formed human with distinct regions and highly interconnected systems. It's the foundational branch that makes all those other anatomical branches possible. 11. Just think about that number for a second. A single microscopic zygote miraculously transforms into 11 integrated organ systems. It is truly awe-inspiring. Every single bone, muscle, nerve, and blood vessel that we study in gross anatomy originates from this intense, incredible period of early cellular division and specialization. Let's jump into Section 3, the third week of gestation, folding the blueprint. If we look at the timeline of early human development, day 1 is fertilization, obviously. And fast forward to the fetal stage, our organ systems are mostly just growing and maturing. But right there in the middle, specifically at the end of the third week of gestation, that is where the real architectural magic happens. During this vital window, the embryo is essentially just a flat disc consisting of 3 tissue layers. Suddenly, it begins this process of biological origami. It's an intricate series of folds that dictate the rest of our physical existence. So here is exactly how that biological folding works. First, the top layer, called the ectoderm, elevates and folds backwards, or dorsally, toward the back. This creates the neural tube, which is going to become your entire brain and spinal cord. At the exact same time, a lower layer folds forward, or ventrally, toward the front. It rolls up to create the gut tube. This dual folding mechanism creates what embryologists call a tube on top of a tube design. You've got your central nervous system on top and your digestive system right below it. The sheer elegance of it is just staggering.
Which brings us to section 4, forming the body cavities, creating the spaces.
As these tubes fold into existence, they don't just create solid organs, they also create the vital empty spaces inside of us. The middle tissue layer of the embryo, known as the lateral plate mesoderm, is a brilliant piece of biological engineering. It actually splits perfectly in two. One half becomes the visceral layer, which wraps directly around the gut tube and eventually forms the delicate serous membranes covering our internal organs, like the heart and lungs. The other half becomes the parietal layer. This pushes outward to form the lateral walls of our body cavities. The space left between these two splitting layers, that becomes the primitive body cavity itself. To seal the deal, those lateral body walls sweep forward, moving ventrally, and they fuse right down the center midline of the embryo. This completely encloses the internal body cavity. Well, almost completely. There's one vital exception. The connecting stalk remains open. This stalk stays attached to the yolk sac for necessary nutrients, and eventually that becomes your umbilical cord. Actually, if you ever want a reminder of this incredible folding process, just look at your belly button. It's literally a permanent scar of your own embryological journey. Oh, and as this whole structure folds closed, the amnion is pulled in with it, creating a nice, protective, fluid-filled sac around the developing embryo. Section 5. When development goes wrong. Ventral wall defects. Up until now, we've looked at the perfect execution of the blueprint. But this highly specialized branch of embryology is also crucial for understanding clinical anomalies. What happens when this intricate folding process gets interrupted? Well, when the lateral body walls fail to fully sweep forward and fuse at the midline, we get ventral body wall defects. One of the most startling examples is ectopia cordis. This is a congenital malformation where, because the chest wall didn't finish closing over during that critical early fold, the heart ends up developing abnormally, located completely outside the protective thorax. No way, right? It's intense. You also see conditions like gastroschisis, where abdominal organs protrude through the wall, or bladder exstrophy. These aren't just random occurrences. Understanding embryology perfectly explains these dramatic macroscopic anomalies. It draws a direct line from a tiny microscopic cellular misstep taking place in a highly specialized branch of our timeline, straight to a complex, life-altering surgical reality in the delivery room. We've seen how the grand, specialized branches of anatomy all trace their roots back to the precise folding of microscopic tissue layers. So, if a single embryological misstep in week 3 can radically alter our entire macroscopic physical reality, what else might our microscopic origin story reveal? Unlocking the deepest secrets of our earliest development isn't just about understanding our past. It might just be the absolute key to mastering the future of human health and medicine. Thanks for joining me on this explainer, and keep exploring the incredible blueprint of the human body.